those bacterial mats or those animals there. Therefore, we don't expect that there will be methane or sulfides in the water. But we don't know until we sample it. And so that's part of it, is could there be nutrition coming in for these guys in some way? But the other thing to look at is where this water is coming from. So we know that it's heated much warmer than the surrounding water. So it's been passing through rocks. It's called a low temperature subflank ridge vent.
Yes, something like that.
Yeah, and so basically water has been percolating through the sediments near here and has been getting warmed up from it. So there was latent heat in those sediments. And by testing the water chemistry, we can figure out kind of the geology behind it, sort of how that venting is happening and where that heating is coming from.
Wow, so we can learn a whole lot just from getting one little water sample.
Tons.
This is amazing.
So cool. Yeah.
So before we leave here, can we get a good shot of where all the equipment is so we know when we come back where it all is?
Also record what our heading is so that our ROV will know how to face when it sets down.
Yeah, if you guys are done looking at the baby octopuses here, I can.
Yeah, you can zoom out.
So we'll start at the top, I guess. You want to?
2 greens at the top.
You can zoom in there again. So you can see the 2 instruments there, the 2 green handles. Hey, I did color coordination. How'd that happen?
That looks good.
Okay, great.
You got
And
those,
then,
do you?
yeah,
Yep.
and then down from there.
Okay,
the
yeah,
next
then
height.
headed down the hill. You can zoom in there.
We've got the blue.
Yeah,
purple?
so you can see
I see.
2 blue handles there. The one, you can actually see all 3 instruments there.
I'm missing the third one. I see the solid blue and then the blue, green, red stripe.
Yeah, you might have to
Oh,
open up the iris a little bit, but I believe the other temp probe is right underneath it.
aha.
That's
Oh,
the
okay.
purple.
So that's the purple sensor.
That's the handle purple.
Oh, I see.
The blue, red, green is the DO temperature probe.
Correct.
And then
Or that's the, I think that's the T handle coming off the, what do you call it, the RBR?
yeah, that's right.
He's the RGB, and then right between the 2 handles there, you can see just the some painted blue. That's the other temperature probe.
Got it.
So
Okay.
they're all 3 in that shot.
Okay.
The other one's, remember I caved in a few rocks on it, so the other one's kind of buried, but it's basically under the RBR and the Osmo.
Okay. Okay, perfect. Thank you. This is good.
And then we'll zoom out and see where the crate's oriented.
Yeah, I'm gonna have to do a little housekeeping here because
Okay.
hopefully all that stuff will stay. I've
Okay.
knocked our O2 sensor off the porch and it's down in the, it'd be interesting to see what the readings on it are right now.
Oh, we can do that.
So hopefully if I just float up here, it will come out of there without
I've
taking
got
the other
no
stuff
data
with it.
points right now.
Ooh.
Am I free?
Yeah, we're not getting any readings back here. It says no data points.
That's not good. So there you can see that. Let me,
Yeah, if we can get a frame grab of that so we see the orientation, that's nice.
let me move that manipulator out of the way.
great. Okay.
So I need to come around and find a place to set your dangling sensor down there so I can grab it up.
Okay, sounds good. Whatever you need for that.
So the other two sensors that are still in the vehicle, we're going to deploy those at another site? Is that
Correct.
okay.
At the Octopus Spa site. And Dan, I'm curious, there was one temperature sensor that was already loose, is that right? The yellow-handled one?
Yeah, it's sitting on the porch next to the funnel.
So without us being here and having our lights on, these guys would be in complete darkness. Does anyone know at what depth would the surrounding water be completely dark? So at what point is it
I
just
only have
darkness
3
in the
beams.
ocean?
Is it that steep here?
Yeah, so there can be twilight, if what they call it.
Yeah,
to
the
what?
twilight zone, 200 to 600 meters is generally
Okay.
the
So,
twilight zone.
yeah,
Two
so there might be a
hours.
little bit of light, but
No.
deeper than that, it's probably pretty dark.
So we define the deep sea as anything below, about 200 meters continental shelf. And so there, it's already quite dark, but there's just a little bit of light that
What's that?
some animals are able to sense. That's why you see deep sea
Roger.
fish with such big eyes at those
I think
depths.
we're thrusting up.
So we've got an itty bitty amount of light, but not a whole lot.
Not a
So
whole
it's
lot.
pretty dark down here. We're lighting it up so we can see what's going
Lot of
on,
depth
but
fail.
they're very used to being in almost complete darkness.
Sorry, I'm just testing one of my sensors now.
So all these particles floating around in the water, are they plankton? Is that marine snow? What are we looking at?
Right now we're probably kicking up a little bit of sediment with our thrusters, but marine snow can be falling as well.
Marine snow is one of those terms that we use all the time because it's really important. It's sort of the food supply to the deep sea. But, what is marine snow? It's not snowfall like what we have on land. Marine snow is
Do I need 4 beams for auto altitude?
is a bunch of stuff. It's fecal pellets
What's that?
from zooplankton that were at the surface. It could also be dead zooplankton that are sinking down. Really anything that
Hey,
was at
Go
the
to the
surface.
sensor page and see if we're getting data from the depth sensor, okay?
Once it dies and sinks, then it becomes marine snow.
All right, keep an eye on the vehicle for a minute, would you? I'm gonna try and do a Roger.
I'm going to pick your brain even a little bit more there with our marine snow. You said there were zooplankton, so those are our plankton species up at the top. Could you just talk a little bit more about plankton and how important they are to our whole ocean ecosystem?
Sure, yeah. So plankton in general are defined as animals that drift with the water or can't swim against the currents. So they can swim, but they can't swim strongly, and so they drift around with ocean currents and kind of circulate around. Actually, you'd be a plankton if you're in a swiftly moving river, but we're generally otherwise swimmers. But yeah, so plankton then can be broken down to either phytoplankton or zooplankton. Phytoplankton are photosynthetic, and they can take sunlight and turn it into food for themselves. And
I
they
won't be able
form
to
the
see anything
basis
with it
of
anyways.
the food web in the ocean. And they also are responsible for about half of the CO2 and kind of photosynthesis that happens on the Earth's surface. So phytoplankton in the oceans are really important for producing oxygen and drawing down carbon dioxide, even from outside of the water, but within the water as well. And then zooplankton
Now you
are
can
not photosynthetic, so zooplankton are any animals that can't swim against the currents and drift in the water. So these are things like jellies, or, gosh, there's so many, or copepods and other
was
small
that for a blind
crustaceans
grab?
that drift with the water.
Can they also be larvae
I was
of
about to- yeah.
crabs or other animals?
Yeah, so while we're used to seeing crabs or clams once they're on the seafloor, before they get there, they have a planktonic stage, and so their larvae will swim up or drift up in the water column. So yeah, plankton are lots of things and do lots of things. They're big for photosynthesis, big for moving nutrients around, and also sometimes are the animals that then become the seafloor animals, and other times are the animals that live perpetually in the water column.
Very cool. So our plankton can do all sorts of things for our environment, for feeding other people, for helping us out.
That's right, and we eat some plankton too. So,
Ooh, yum.
yeah, well,
Porch is retracted, right?
No?
the krill oil extract is, a supplement people take, and that comes from krill, which are plankton, that live in the ocean, and actually those are
Okay.
what primarily feed whales.
Back in action. Housekeeping complete.
Excellent. Then I think we're ready to move on to the octopus spa location.
Yes,
Agree,
we
Erica?
are. There's one sensor we don't have any data for. I don't know if we should try to fix that or move on.
We
Yeah,
can reboot en route.
Okay, great.
We can do that en route.
Okay.
So,
So yeah, this looks good. I'm glad we got some good overhead shots of this. It'll help us refine it, if that's a word. Refine it.
Refine.
Find again.
Scavenger
yeah, I
hunt.
can get some different shots now if you want. We got all our toys put away, so.
Yeah, as we're lifting off, it'd be nice to look down the gullet of that.
This looks good.
The gullet.
Bridge nav. Yeah, 20 meters, bearing
Curious
005.
how the octopus move these things about, if at all, when we come back.
They'll rearrange it, make their own little octopus garden out of the things we've left them.
That's right.
They
The feng shui is all off.
The feng shui, that's it. They're waiting for us to leave so they can readjust everything. I know we don't know too much about this species, but there are other species that do that, correct? They'll arrange rocks or shells or things and kind of build themselves a cave or a garden.
I think so. I think some tropical species will do that a bit. Kind of make their own little hollow.
So we just head up the cliff to the north.
Yep.
Yes.
Copy.
It's curious, like these brown spots that Amanda noticed, we thought maybe they might be old cement from the octopus, but we're not sure.
I'm now
They seem a little off. They're not right at the vent.
I'm thinking now, because once we got up, I saw a lot more. Oh, look at those. That neon yellow highlighter sponge. Cool. I am so curious who that is. We collected one of those last year and haven't figured out who it is yet.
All
It's my
right.
puzzle. Call it Manda's mystery sponge right now. Sorry. I lost all
I'm gonna
train of
kind of
thought.
follow the
The
cracks
cements.
for a while before we turn on our true bearing.
Okay. That sounds great. Right, about the scratches. I'm thinking they might be scratches from the ROV because I saw a lot more of them once we lifted off.
Oh.
So that rock is so friable, maybe we're eroding away the top layer and seeing some of the muck inside. Ooh,
You want to zoom in there for a second?
there's a female who's doing that ventilating motion. She's
Oh, that's that
swashing
movement
inside.
of like sort of wiggling around. She's oxygenating her eggs.
Yeah.
Oh, cool.
And Erica, do you know what anemone that is?
I do not.
Well, mystery anemone.
But when we zoom back out, there's a
Pussy star.
acorn
Zoom
worm on
out
our
of it
left.
there, yeah.
See, that's that
Uh-huh.
purple organism. That's an acorn worm. It's a hemichordate. It's actually more closely related to us than these anemones or cephalopods. octopods, which is weird to think
Which
about.
is weird.
Okay.
Great,
Moving
thank
on.
you. Oh, there's mushroom corals to the left.
Mm-hmm.
Ooh, there's an upright octopus.
Oh.
I wonder if that's a male. Can you do a quick zoom? It looks like it's got its third arm out, so we'd be
Yeah,
able
go
to
ahead,
see if
Aaron.
it has
Zoom
that
it.
modified
That's
hectopodalus
good there.
appendage. We've got arm number 1, 2,
It's the one curled up,
yeah,
facing us.
I think so. That looks like arm. Oh yeah. Yeah, so that's a male.
You can
You
tell,
can-
huh?
Yeah, you can see it's the third arm from the right eye. So the right eye, that's arm number 1. To the left is arm number 2, and arm number 3 is that backmost
Oh, in the
one.
back. Oh, yeah,
And
yeah.
it's got a weird,
It's sort of straight right there.
like a pad. Yeah.
Mhm.
Okay,
So
and you
that's a
can
male.
zoom in a
So
bit
this
more
is
there.
a male octopus. A lot of the other
Float up there. That's good.
octopuses
Thank you.
that we have been seeing
There's the
have been
hectopodalus.
female, and we can tell from this third arm that this is indeed a male.
Good eye, Amanda.
Thanks.
Nice. He looks like he's posing.
So we watched
He's like, I'm cool.
2
Yeah.
instances in August of a male octopus approaching a female, and then it would rear up on its son. Oh, what are those? Those pharaonids? Sorry. They're little worms to the left. I think they might be, well, they're maybe just a type of polychaete.
I don't know.
Could we do a quick zoom on that too?
The worm to the left of the lasers?
Yeah.
Or just above the lasers there?
No,
To
to
the
the
left.
left. They look like
They're-
feather
they're
dusters.
Very
Oh,
transparent.
I see. Yeah.
Oh,
Okay,
good
I
eye,
can
you
zoom in a
guys.
bit there. It once did want to drift left. I'm touching somewhere. These guys here?
Yes,
Yeah.
those guys. Huh.
Those are like a feather duster type worm.
Yeah, I think so. A sabellid.
So those would be a coral?
A worm, actually.
A worm?
Annelid.
And it lives in the tube.
Those look like pharaonids. Don't
So
they?
tube worms who
No,
would
I
stick
just
that
really-
out to kind of catch
Oh.
food as it floats by?
Yes, that's right. Can you
Very
also zoom, please, on those worms that are just between the lasers, or just to the upper right of them now, on the rock? To the right with the sort of orange tubes.
Yeah, go ahead, Eric.
Oh. Great. Aha. So that's a different kind of worm. That's the one, I'll remember it in a moment, that has the little plug operculum thing.
Well,
Oh.
we're hitting.
Okay.
Serpulid?
Might be a, yeah,
A
serpulid.
serpulid. So there's a variety of worms here. That's what we're seeing. And those are also filter or suspension feeding worms. Okay, thank
All
you.
right. Thanks.
Moving on.
So these worms like this habitat because they can pick up a lot of food out of it, right, as it moves by through the water?
Yes, that's right. So as the currents bring water past them, then any edible particles can get stuck against the mucus or the or little hairs that are on those projections. Right. So I was saying the males, we observed the male to rear up on its back arms and sort of flare up the underside of its body toward the females, and then it would reach out and touch a female. And both attempts that we saw at contact were not welcome, and the females kind of brushed them away. And then the male would go off on its way to try to find another female that was more receptive to it. Or, okay, I shouldn't say that. I have no idea what it was going off to do, but it didn't stay there. Ooh, look at the size of that sponge.
Ooh. Very cool.
Can,
That one will pump a lot of water.
Do a laser zoom there if you want.
Sure. Are we
Yeah,
are
we're
we
good.
on a heading toward the spa now?
No, still kind of following that crack.
Okay.
And the little, see the overview of Argus where the little white, following the white dots?
Okay.
You want to do a quick zoom on the sponge?
No, that's sufficient actually. That's good.
Okay.
Thank
And
you.
as a reminder, how far apart are the two laser beams?
10 centimeters.
10 centimeters.
So that's a nice sized sponge.
I didn't see.
Wow, what an incredible habitat
It
this is.
just stopped. We moved 40 meters, and so you still got some swim.
Yes.
So got our
Oh, there's a little
octopuses.
squat lobster up to the upper left.
It's like the B-52 song, Squat Lobster.
I think that's what that is.
If you guys want to zoom, just you can, as we're coming up the cliff here, you can have Aaron zoom at will.
Sure thing, thanks. Can we also cycle power to that temperature and DO
Oh,
probe?
yeah. No,
Hmm.
it's not any of those. It's a 12 volt.
It was on that page. No, it's the same as Herc's oxygen sensor.
Oh yeah, so you guys will lose Herc's oxygen sensor for a minute as we cycle power.
Okay.
Are those bathypathies? No, that's not right.
Those might be umbellapathes.
Umbellapathies. Beautiful.
Another black coral.
Coming up with me? Yeah, there you are.
And those white squat lobsters were Munidopsis.
Got a sea cucumber over to the right. Oh, I think that's a benthic,
Where are you looking?
ctenophore, right in the middle.
Oh, and it's attached to
Down
something?
attached to the rock.
Mm-hmm.
Dan, do you see that small thing with a red dot in the center to the right of the screen on the rock?
Right center. Now it's coming to
center.
center.
So that's the purple to the right of that.
I see it, yeah.
I believe that's a benthic ctenophore, but it would be nice to take a look.
You can zoom in there.
Yeah. So that is a benthic ctenophore. So ctenophores are also called comb jellies, and we're used to seeing them in the water column as swimmers. But there are some ctenophores that can live on the seafloor. So these guys will attach themselves to a stalk, as you see there, and then they'll put out tentacles, which you see drifting to the right of the animal, with projections called tentilla on those. And those tentilla have sticky sort of batteries of, they're not cells, but they're structures called colloblasts. So those colloblasts make this sticky substance, and they can stick to prey that touch those. So, thank you. That's a really neat find. We're good to
Okay, I
go.
hate to do this to you guys, but I need to spin counterclockwise to bring the ROV
Okay.
starboard. Some tether management here.
That's okay. There's that giant cuskeel in the background, one of my favorites. I like to call him Spunky because his genus is Spectrunculus. I call him Spunky.
Spunky the cusk
You
eel.
want to come up 4 meters for me while
Oh.
I'm doing that?
That's a grenadier. That looks like Coryphanortes armatus, which is the abyssal grenadier. It sometimes can be confused with Yakine, and I might be pronouncing that wrong. But for now, I'll call it armatus.
How do you
So this is
differentiate
its
them?
fish.
We need to have them in hand, and I don't know the exact answer to that, but they're hard to differentiate in video.
Are there other rat tail or grenadier in the area that are more distinct?
Yes,
Now go
there's
straight.
one called the ghostly
Right.
grenadier, which is mostly white on the head and modeling towards the tail, and it has a funny-looking nose. Funny to me. And then we can also get Philipher down here, which has long pectoral fin rays.
And now
They're the
these are
pet-
related more towards our bony fish and things, or are they the
Yes,
chimera-like?
yes, these are bony fish. They're related to cod.
Gotcha.
Erica, sorry, because I'm not a fish person. How do you recognize a cusk eel then? The giant cusk eel, because they're also pointy with a thin tail.
Yeah, I can sort of tell by their head, but they also have barbels under their chin that they use to feel for food, and sometimes you'll see those on the cusk eel. But a lot of head and then, sorry,
You want to come up a bit more?
a lot of
So it
tail
looks like
compared
I'm
to
going to
the
have to go
head.
under you.
Ah. Okay, by the end of this cruise, I'm going to learn how to identify fish. Those fish. Okay, not all fish.
You'll get those.
That sounds like a great goal. Were those guys that we just passed on the left sea cucumbers?
Yes, the bright pink.
Mm-hmm.
Yeah, those were sea cucumbers.
Very cool. What do they eat?
Oh, they're deposit feeders, which means they eat stuff that's on. See all that stuff on the rock? It's covering the rock. They eat that. So sea cucumbers will really specialize in eating sediment, and then in their intestine they extract all the usable nutrition from it, and then they poop out the cleanest sand you'll ever find.
Lovely.
And you can see sea cucumber fecal casts because they come out in sort of little tubes or spirals. And so we might see some sea cucumber fecal pellets as we're diving.
They have like a unique structure to them. That's interesting.
Yeah.
We'll try to point some out next time we see some.
Yes.
Good call. I know I've talked quite a bit about poop so far between sponge poop and sea cucumber poop, but it's because, especially in a habitat where there isn't a lot of food like the deep sea, recycling is really important. And so, you know, food serves its purpose once for animals, but there's still usable carbon left over in the fecal casts that are left behind. And so carbon and food can get passed around from animal to animal, and it's really important to know how that kind of moves around to understand how all the different species here get their food.
Yes, it might seem weird, but it's an incredibly important nutrient source for lots of different things.
There's several different sea cucumbers in this shot.
Like purple ones and
Yeah.
pink ones.
Dark maroon.
Beautiful.
There's one that's rearing up. Isn't that a reproductive thing? Is that right?
Ooh, I don't know.
I know we have talked about it on Ambari trips. And
I thought they were
I
looking
wanna
for
say it
food,
was-
but that sounds like a more interesting posture.
So some neat feeding structures that sea cucumbers have are oral arms. So around their mouth, they have these little arms that stick out so that those arms can kind of grab on to pieces of sediment and bring it up to the mouth. So they're not just sort of chomping at the sediment, they're grabbing it and bringing it up to their mouth. And in some sea cucumbers, I haven't seen any yet on this dive, those oral arms are instead very frilly with very fine processes, and they stick those arms up into the water. And those sea cucumbers are not deposit feeders. They are now suspension feeders, and so they catch particles that are drifting on those arms.
I should be able to look and go north of it now.
Okay, great. Thank you.
Our loggers have come back. Excellent.
Oh, fantastic.
We've got oxygen and temperature again.
Reboot complete.
Perfect.
So I've got two questions coming in. The octopus that we are looking at, do the guys have their chromatophores, or why are they so light? Is it because they're in the deep? What do we...
Have we seen them change color? I haven't.
I have
we
not.
So I don't have an answer to that.
We're not sure.
Yeah,
That's
we're
a
not
good
sure.
question.
It is a good question.
It is. We haven't seen them. Even the ones that have decided to help us with our science, I don't think we've seen too much of a color change.
If we had touched a shallow water species, we definitely would have seen something. And we've been touching these, not on purpose, and we haven't seen a color change. So that might be our answer.
That's true.
Oh, that's interesting. And would that be because they're so deep that they wouldn't need to have sort of that camouflage or color change because everything
Well,
down here is
you know,
pretty
you can't
dark,
see anything down here,
or-
so it may not be energy efficient to have those if no one is viewing them.
Wow.
There are some organisms in the deep that produce light, bioluminescence, and I don't know if the octopus does. Some of these cucumbers do, some of these corals do. My first assumption is I bet the octopods don't produce bioluminescence, but we've been wrong on other organisms, and they do.
It's true.
Seems like almost all things do down here.
Vampiroteuthis has bioluminescence.
Okay.
So, yeah. It's possible for cephalopods too, but whether these guys do, I don't know if we know.
Just don't know yet.
Yeah.
And it probably isn't a good idea for us to turn off all of our lights down here.
Yeah.
So we won't know unless we were to bring it up.
The cameras also aren't sensitive enough to pick up
Looks promising.
bioluminescence.
What's that?
There's a whole aggregation over there which is in the general area we want to be.
Octopus Spa had sort of 2 long parallel bands of octopuses in a cobble field.
Yeah.
Are you seeing something like
That's kind of
cobble
what I see
field up
to
ahead?
the right, just off the ridge.
Roger.
While we're searching
So are
around
we
here, are
close to the waypoint
there
already?
any invasive species in this
Oh, I see
area?
it. Good eye, mouse. Thank you.
I don't know of any invasive species in the deep down here.
Here's Octopus Spa.
Navigating by
Oh,
camera
there
again,
they
I see.
Yeah.
are. Oh my
The
goodness.
deep sea is very well, it's a lot more connected or homogenized.
It was in the right area.
So, so it seems like we find species with a much greater distribution here. So I don't know of any invasive species that we recognize as invasive in the deep sea.
Wow, that's a lot of octopodes.
So this is what we're calling the octopus spa, correct?
Correct.
This looks familiar?
This does.
Good.
I took oxygen samples here.
I think the last time we came here, we noticed a 10 degree, almost a 9 degree or 10 degree difference from ambient. So that's why we call it the spa, because it seemed to be warmer than what we had observed previously. But it may not be the only place where it's warmest.
Yeah. We saw 9 degrees here and yeah, 10 at the fissure, the vent. So
Okay,
yeah,
so
these were our nice hot spots. And you saw how close they were. I think, Miles, I heard you say we were 20 meters away? Is that how far we were?
Yes. Oh, from the target, but it looks like we're almost looking right at it. So,
Well, we can look around a little bit and see if they get a little denser. I don't know how big this was. A football field, Chad said?
Yeah, Chad said. I only remember visiting the one spot. We kind of stayed in this narrow band.
Okay.
And just so you guys have an overview of the sampling we'll do here, we want to do more of that in situ temperature and oxygen measurement around and among the octopuses. And also take 2 to 3 squeeze samples, the squeeze samplers, and we'll leave the other 2 temperature loggers here. That's sort of the suite of things that we want to do in this area.
Okay.
Before setting down, maybe we can do a little reconnaissance just to see how large this is. Does that sound like a good
Yeah.
idea?
I think so. I can see an Argus a little bit, but is Argus's view, it looks like it's a furrow or like an indentation. Is that accurate or?
yeah, sonar's showing that too.
Okay. I wonder if there might be more farther down. You said we're not quite at the waypoint yet?
No, we're within 20 meters, which is, at this depth, pretty much spot on.
Okay.
But if this doesn't look familiar, then we can search around.
Well, this is my first time here, so
Yeah.
I'm going to lean on
Little
Amanda.
bit.
Well, I'm
I
going
remember
to
it being a little light.
come around and
This is not a football field.
It's not a football field, but I remember
No.
there being sort of 2 long fingers and a area in the middle that we could sit, which I do see here if you were to turn the ROV, looking at Argus's screen, if it turns 90 degrees to the right.
yeah, that's kind of what I'm seeing. You can see on the sonar kind of the little bowl there.
Mm-hmm.
So if I come down and sit in that bowl, everything will be uphill, which will give us a better view.
It's probably this spot then. I just have a different perspective of it right now.
It seems like
It is a rubble field. There are
is.
lots of octopus.
It's true. I think this is a perfectly good place to put the other temperature sensors.
There's a good kind of landing field right there in front of Herc.
Great. Let's do it.
I can, with the little mini spot, will be off to our right side there.
And we're going to want to do the temperature and oxygen. So perhaps while we're setting down, it would be good to look for wavers in the water, the shimmer.
There's a sponge.
There's my yellow sponge.
Yeah, I won't set down yet. I'll just get closer and kind of skim around.
Okay.
So we're looking uphill now, so you're getting some better video, because I can't really look down unless you want to see your samplers.
Oh, definitely. This looks great.
Look at those fly trap anemones.
I'm not seeing a lot of shimmer yet. Could we zoom a little bit more, please?
Sure.
I see a little bit in the upper
there's
right.
some shimmer.
Yep,
Yeah.
yep.
Oh, there's shimmer.
Okay, shimmer.
Shimmer present.
I can pan down a little now that we're zoomed in. Ah, not that far.
Yeah, there's quite a bit of shimmer right there. It's probably going to be a really good spot. Do you think that that's a spot where you would be able to set down and...
Yeah, that's a
those
good
probes in?
spot. You can see in Argus, we're not going to sit down on any ladies, so.
That's great. Okay, let's sweep a little bit more to the right just to get a view of the rest of the octo field, and then let's return to this heading to set down.
Sure.
We see lots of lady octopodes, and we see a number of different anemones. Do we know if there's any correlation? Is it just a similar environment that they both like, or is there something going on between the two species?
I would imagine the habitat is providing something that they like. I don't know if they're operating together.
Same.
They may have some, the fluid coming out may also provide cleaner rock for the anemones to attach onto.
That sponge is Hexactinella species 3.
Where am I looking? Oh, straight
To the
ahead,
left of the
white,
lasers.
The white one?
the white one? All right.
Sorry, I know, our naming conventions. So at Mbari, when we don't know what a sponge species is, but we recognize it morphologically, or if it hasn't been described yet, then we have to call it by names like that. So that's Hexactinella species 3. Oof.
And I think these anemones are from the family Hormathidae, and they get into a posture that they look like a Venus flytrap.
They do.
Not all the time, but sometimes.
So we can sit anywhere in here?
Sure.
You want to
Sure.
be back in the thick of it, do you? Or? Feels like shot here is.
Okay.
I got one on the bottom left that was ventilating, or venting her eggs. Everyone else seems to be pretty calm. We have a question of, are there hydrophones on our ROVs? What would it actually sound like down here? So I know we're a little bit noisy, but have we ever heard what we sound like?
Yes. You know that scene in Dumb and Dumber where he asks, what's the most annoying sound you've ever heard?
Uh-huh.
That's it.
Oh no.
It's the sound of the HPU that powers the arms and the thrusters on Herc.
Sort of like an engine type sound.
It's just a whirring, yeah, a
Gotcha.
loud electric whirring.
Sound of a 20 horsepower motor driving a
2,800 psi
I forget
hydraulic
the
system.
exact PSI on our pump, or the
Well,
displacement. Okay, Herc has landed.
All right. So if we can put
Poke
the
around
temperature
with the temp.
probe around one of these places where it looks like it's shimmering.
it's
we
already
can't.
come up 0.2 since we came into this little area. Or at least
There's a hydrophone in Monterey Bay that is, how deep is the Mars cable? Over 1,000 meters.
Wow.
And you can actually hear rainfall all the way down that deep with the hydrophone. It's quite amazing. And we hear little earthquakes and ships going by, and the ROV if it's nearby, and whales, whale songs. But the rainfalls surprised me. It's like, how can we hear rainfall all the way down here?
That's really interesting because that sound travels so far through water.
One of our
Yeah.
hitchhiker.
Is that something the public can listen to?
Yes, there's a listening library on the MBARI website. So if you Google listening library MBARI, you can hear live sounds, and then there's also recorded sounds to familiarize yourself.
Wow.
And that resides on the Mars cable. It's an observatory that scientists use to plug in power to their equipment to do several different types of research in the deep.
The person wanted to know the sound of the environment, but not the ROV. Well, it would be nice to hear the environment, but with our whirring of engines, it's very difficult to just kind of hear what's going on without us being there, so we kind of interrupt that sound. But if you go check out that Mars cable, it sounds like you can learn a little bit more about what the ocean bed might sound like without our engines. So for you who are just joining us here on Nautilus Live, we have come to our octopus spa. So we call it the spa because it's just a little bit warmer than our surrounding areas. So you might be
Give me
able
a
to
quick
see some of
zoom
that
there.
shimmering or seeps.
Thank you.
And that's that slightly warmer water coming up. And you're viewing a whole field. I don't know, are they clutch? Are they a patch? What's a group of octopus called? We've got our group of lady octopus here that are brooding. So they are all protecting their eggs and taking care of them until they hatch. We are not exactly sure how long they incubate or when they will hatch. It seems like this environment is somewhere where there are always octopodes, octopuses, multiple plurals, to check out this area and kind of see where they can lay their eggs. So it's a rotating environment, but there are a lot more here than we
Okay,
seem to
give
find
me a
elsewhere.
laser zoom, please. Perfect. Eeny, meeny, miny, mo.
So this is our temperature probe.
So we're just going to do a little science brief. We'll be back in a minute.
Can you show me the minute with bubble cam?
So all of our octopuses that we are seeing here are octopodes. We have lots of different plural names for our octopus. It's a constant debate. It is a Greek word, but we've tried to put a Latin ending on it, or we have put a English ending on it, or we have decided that we should stick with their family or their order name. excuse me. So they can be octopi, octopuses, or octopodes. So we have lots of different pronunciations of the plural of octopus. And while we're looking here at our octopus, do some fun facts. So they are cephalopods. The word cephalopod actually means head, foot. So they have a big head and lots of different feet. So they have kind of a fun, quirky name. So it looks like it's just about time for shift change. So as our scientists talk, let us know what questions you guys have about our octopodes and our environment. And we'll get set up to talk with our next group.
So the temperature was going up there. That was a pretty good spot. Our sensor samples every 5 seconds. And so it takes a little while to integrate over time. And so it looked like it got up to 3.5 degrees where we were sampling there, which is pretty consistent with what we saw when we came out here with Mbari in August. It was like 3.5 to 4 degrees, although Chad was able to get temperatures up to 9 degrees in this area. So it is possible.
Hey everybody, just a few more minutes as the watch change continues over and we'll get settled in to continue the dive.
Well, we almost got another one. That's why you moved the sensor. Video, we have partial zoom on Argus? Okay.
Do you want
Yeah,
me to
just
pull
come
out just
full
so you
wide
can
and we
see?
can just see where.
Yeah.
Okay, yeah, so we can go back in.
Cool. Thanks.
Okay. Is that a good height for Argus?
Yeah. Science is busy back there.
Huddling.
Height 12.
Good morning, everyone.
All the temp probes match.
Herc, are you waiting for science direction?
Yep.
Okay.
Video, push all the way in.
Ooh.
These anemones are great.
Is that one of those mini lobsters we're supposed to get?
I think it's just a little regular shrimp. I could be wrong. It's certainly happened many times before. It's pretty awesome last night. So our watch started the blue water. We started the descent for this, and then we went to sleep and sort of woke up, you know, feeling the ship still on station. I was like, we're sitting on top of a bunch of octopus. You know, you got to like get out of your bunk, come up to the closest screen, and realize that this is what's happening and had been happening since we got down to the seafloor last night.
All right, I think the shift change is complete.
Okay.
All right, Aaron, let's come full wide on Herc.
All right, so at this site, we were just being briefed by the previous shift. We're going to eventually want to deploy 2 temperature loggers here, take the remaining squeeze samplers, fire Niskin, etc. So for right now, this is great, just kind of leaving that probe in various cracks and crevices in this area, letting it sit, seeing what kind of tempers we can get. And if we get high enough, we'll deploy some of that other equipment. We might want to at some point, I'm just curious, the differences between this probe, because the sensor's in, you know, kind of buried in the shaft near the
versus ours.
Versus yours, yeah, where you can kind of get it into cracks and crevices a little bit more. It'd be interesting to see that. And then also, since we're going to be here stationary for a little while, Dane, do you want to fire up the probe?
Yeah, I'd like to do a test run. So if we can get that bubble cam focused on my telltales.
And do you still want to record?
No, not here. Oh, that's yes.
Dane, will this test run count as the start of your sample?
No.
No.
It's going to be a very short period of time.
Okay.
So
copy that. Thank you.
Okay, so when you are ready, go ahead and turn the pump on. Give us a countdown for that.
Okay. We'll turn on at 1, 5, 4, 3, 2, 1. Pumps on.
Beautiful.
Yeah, that one telltale.
Yeah, pushed out.
Yep. All right.
Okay, I think we can, well, let's leave it on for just a few more seconds.
So
Aaron, let's push in.
This pump is going to be running in the background, just continuously
Hold that.
pulling in and filtering water while we're conducting the rest of the sampler deployments and operation.
Right.
It's a great view of the shimmer. Nice shot, Aaron.
Okay, Greg, I think that's good enough for the pump test.
Pump off.
Yep. Perfect. Okay. Thank you.
So welcome back, a few viewers joining and surprised by the number of octopus we're seeing. This is a spot, the octopus garden.
No, it's not
octopus
actually.
spa, as we're calling it here, the specific site. We're on the site of Davidson Seamount in Monterey Bay National
Where are we getting
Sanctuary.
a readout? Sorry.
No worries.
Where are we getting a readout from this probe?
I'm not sure if you guys have access to it or not. Justin set up a web browser tab that displays it. I don't know if you guys
What's
have
the
access to
page?
that or something
Or
else.
what
It's
screen are you on?
hold on, let me just come up there.
I can just change the screen.
What do you?
Like, are you viewing it?
Sure. You can do data.
Okay, it's on. Wait, the data, is that a screen?
Capture.
Capture, got it.
Actually, you might be better off with Chief Sai because I'm going to be periodically taking notes. Chief Sai will probably be more stable.
Okay, and which one of these, is this all of it? Is this all from this probe?
The only part that's from the probe is oxygen concentration psi and oxygen saturation
that middle row.
psi and probably
Optode temperature.
optode temperature psi. So the middle row.
Middle row.
Yep.
Got it.
And
We'll
the
take a
rest
look at
is Herc related data. You'll see oxygen concentration OET below the psi row. That's not the corrected value.
This looks like the temperature is very slowly increasing.
Yeah. It's pretty cool there in meso.
So is the temperature probe that the entire metal end of that device?
That is actually a plastic tube,
Ah.
and then it's a optode. It's the same probe that was left on the deployment.
Okay.
So it's oxygen and temperature.
Got it.
So lots of the question, why are the octopus here? And that was part of what we are investigating right now. Last year, discovered the site. Surprised, delighted to find over 1,000 of these octopus in this breeding position all along the flanks here of Davidson Seamount. That was a new discovery and surprise, as well as the shimmery water that there is venting. Low temperature venting is what we call it, but that's only compared to like the mega black smokers that we call high temperature venting in the deep sea. We're looking at about 10 degrees Celsius as the highest, which is you'd want to wear a wetsuit or a dry suit if you were swimming in that, but compared to 1 degree or 2 degree, which is the rest of the water at the bottom of the seafloor, the deep sea,
That would be a good spot.
this is kind of a toasty area. So we have lots of questions. Does the warm water bring the octopus? Does the venting fluids, chemistry bring the octopus? Does the clear rocks bring the octopus here? Does something else entirely? Maybe there's an attractant of
All right, it looks like
prey.
temperature's dropping, so I'm going to try moving to a different spot.
Roger.
Yeah, we don't know the answers to any of those questions yet, but that abundance of questions is what made us want to come back here.
Aaron, full line.
So here's a question. Why would the temperature start dropping?
Sometimes you just get like a current flow. If you look in the side of this, that opening is where the sensor is. And so it was, I guess the idea was that the venting fluid would go up the tube and then kind of exhaust there. But if you had a current that blew in that way, you'd get a temperature read in the wrong direction.
Okay.
Or if you like bumped a rock.
Yeah.
Yeah, or you bump a
the
rock
mixing.
and you change the flow.
I'm wondering if anybody knows whether flow is surgy, if it's responding to water pressure waves passing above it, or if it's constant.
I think our best indicator would just be to watch the shimmer for a while, but I would think that just